So the limitations of this talk are first, that I'm not a clinician and I'm working with mice. And a second limitation is that among the core concepts of ASD diagnoses, social deficits, repetitive movements, and language deficits, in our experiments we're only dealing with social deficits. Now I think that that is probably the core concept of diagnosis, but another advantage of the work that we're doing is that we can deal with cellular mechanisms, which be neuroendocrine and molecular endocrine expertise that we can bring to bear on the question of sex differences in autism. So by way of background, let's talk about sources of male vulnerability with respect to symptoms of autism. But I also offer the possibility that we're talking about female protective mechanisms, and I don't think that those two things are mutually exclusive. And you'll see them on a total of, this heading on a total of 4 slides. And the reason is that there's a small number of people, the ones that come to mind would be John Constantino at Washington University of St. Louis, and also Stefan Sanders, who I think is at UCSF, who keep talking about female protective mechanisms. But I have to say that I haven't seen any genetic evidence for it. Secondly, Sylvie Goldman, one of my collaborators, thinks that there may be a diagnosis bias. And thirdly, I just read a paper by Daniel Messenger, who's at University of Miami, who did a rather complicated behavioral experiment with children, but who said, the last sentence of his abstract is that he said that he could not find any evidence for female protection. So even though I'm constrained to put it at the top of these slides, I don't really understand it, and I'm not actually sure it exists. Now, what I'm going to concentrate on, being a hormone action kind of guy, is mechanisms that derive from the Y chromosome that the male has and not the female, where we're talking about the effect of the SRY gene on a cascade of genes called the SOX cascade, thus to develop the medulla of the indifferent gonad. Remember, the gonad can turn into testes or ovaries. If you develop the medulla, then you're going to have testes, and you're going to have testosterone as a hormone and a prohormone. And what I mean by prohormone is that testosterone, when it acts in the brain, can be effective because it is transferred into other kinds of steroids, like dihydrotestosterone and, amazingly, into estrogens. So this is the source of my expertise, and it's what I'll talk about most. But in fact, it's only one of 4 possible conceptions of how we get sex hormones, how we get sex differences in behavior. A second source of sex differences would be simply the number of X chromosomes. Obviously, the male only has 1. The female has 2. And if each of these little guys is a neuron, you can have X inactivation in some of them and not others. And furthermore, the mode of X activation in these little fake guys, the red would be females from the XP, and this would be from the XM. But the ratio of neurons in any given cell group could either be balanced or it could be skewed in one direction or the other. So this gives source to tremendous variety of behaviors in the females, which may be important. Thirdly, and a little bit more complicated, is parent of origin allelic imprinting of specific genes. Imprinting of a gene on the X chromosome that came from the father, the paternal X chromosome, leads to, this is the data, leads to expression of that gene in both sexes, but only in males is the gene expressed in all nerve cells. And this would be the data of Barry Cavarin, who's a professor at University of Cambridge, who worked on this beginning in the 1990s. Imprinting of a gene on the maternal X chromosome leads to expression of that gene in females only, but not in all of the neurons in the female brain, but only in some of them. Again, a complexity that people weren't anticipating even 25 years ago. Finally is a new hypothesis, which doesn't have a lot of evidence for it yet, but it appeals to me a lot. The males do not have an inactive X. They're using the X that they have, the only X that they have. Whereas females will have inactivated, in most cases, one X chromosome or the other. And this X chromosome that's sitting over on the side of the nucleus not used is possibly soaking up epigenetic factors that are active, that are supposed to be active, on the other chromosomes. So those epigenetic factors could be things having to do with DNA methylation, things having to do with histone modification or microRNAs. And any of these could be soaked up on one of these inactivated X's, either XM or XP, such that they're not available in the concentration that they're supposed to be elsewhere in the nucleus. So this is an understatement, actually, of the complexity of the situation, which will allow me to give you a couple slides that my secretary, who's a professional artist, made for me something more than 10 years ago. And this is the old view. Let's call this the 19th century view, and we'll talk about the new view in a minute. The 19th century view would be that there's a baby at the top of a mountain, and since I gave this talk in Stockholm about maybe 12 years ago, it's going to have to be a Swedish baby. And as that baby skis through life, the baby only has two choices of gender roles in the 19th century, strongly masculine gender roles or strongly feminine gender roles. Whereas the new view would be that even 12 years ago, we had this number of intersections where things can change. And now, if we added all the data up to 2015, the number of intersections would be much larger. And in a case like this, the new view is that as the Swedish baby skis through life, not only are there strongly feminine and strongly masculine gender roles, but there are lots of other ways for the baby to reach the bottom of the hill. This, for example, would be testosterone and anti-Mullerian hormone. We have critical periods, neonatal and pubertal. And finally, there are straightforward behavioral things that dictate gender roles. For instance, if you're in a small town in the middle of Iowa, the question would be, are there any cute girls in town? Or are there any cute boys in town? So matters of social affinity are going to be just as important in some cases as all of this biochemistry that I've been talking about. The sources of male vulnerability other than genetics are going to be environmental mechanisms. I happen to favor social stress and social environment, but there's also maternal immune activation, and there is the paternal age effect. And finally, it will be the hormonal mechanisms, and that's the source of my expertise. And so naturally, I tend to concentrate on that. What I did when I realized that there was this huge sex difference in autism, I thought, and this is like years ago, I thought that's something that we can wrap up real fast. Autism is really complicated. There's the heterogeneity of autism. But the fact that it's 80% boys is something we should be able to explain. So what I did was I got on the bus, and I took the bus up to Albert Einstein College of Medicine, and one of the great pediatric neurologists in the United States is Isabel Rappin, who just retired, and her postdoc, who's developmental psychologist Sylvia Goldman, would spend week after week going through the literature and figuring out what kind of sensible theory of the sex difference in autism we could hatch that was consistent, with the facts that we could read, and also which would make predictions. It's very important for a theory to make predictions. String theory, for example, that you read about in physics, has gone 30, 40 years now, and it hasn't made any testable predictions yet. And so some people think string theory will die out the way that the ether died out at the end of the 19th century. Well, our theory hasn't died out because we do have testable predictions, as you will see. So it starts with the androgenic hormones. Androgenic coming from the Greek word meaning male generating. And testosterone is the famous one. And it turns out that some wonderful neurons in the lower part of the brainstem, in the brainstem reticular formation just above the spinal cord, called nucleus gigantocellularis. These, as the name implies, these are some of the largest neurons in the brain, nucleus gigantocellularis, NGC for short. And these NGC neurons express androgen receptors. And we think that to make this into just a phrase, that these NGC neurons are the most powerful and essential cells for supporting the most powerful and essential function in the brain, which is to be awake. If you have general arousal equals 0, you're either in a comatose state, you're deeply anesthetized, or you're in deep stage 4 sleep. If you want to get out of any of those, you need nucleus gigantocellularis. We theorize that as a consequence of androgen action there, we have greater activity in ascending arousal pathways, coming all the way up indirectly to the amygdala, and here we have the exploded amygdala up here, and increased deposition of arousal neurotransmitters such as norepinephrine and dopamine in the amygdala. And the work, the really well-known work of Benno Roosendaal and James McGaugh at University of California, Irvine, were the first, those works were the first to show that increased deposition of these arousal transmitters in the amygdala is very important for fear learning. And that's important for us because we're going to be talking about social anxiety, the social anxiety of autism. So the phrase that I used was a supersensitive amygdala. And actually, at the most recent neuroscience meeting, much to my joy, I saw 2 posters directly supporting this idea. So a factor, one of the 3 hits of autism, the testosterone, being a male, that's one factor. But a second factor that we've hypothesized is neonatal stress, prenatal or neonatal stress. And I call them social stresses because they're always associated with the mother. One of the most famous ones for generating the autistic phenotype is maternal infection. And we think that the testosterone action with a supersensitive amygdala in the presence of social stress is going to lead to overreactions to stress, social anxiety, and eventually the social avoidance of autism. Let's call it the what's it called when they don't pay any attention? Aloofness. The aloofness of autism. I think that's actually a very well practiced avoidance response. What I mean by that is that if you teach a rat to avoid a shock to its foot, and it can press a bar to avoid a shock when you give it the conditioned stimulus, at first the rat will go nuts. The rat's extremely upset, and reasons I won't go into all the details, but eventually it learns to press the bar and avoid the shock. A well-practiced avoidance response is, let's say if there's a 30-second interval between the conditioned stimulus and the shock, the rat just hangs out, it's very, very cool, like an average Californian, and then it comes up, and there's going to be a shock pretty soon. It just ambles over to the bar, and it presses the bar. I think that at least some of these kids who avoid social contact are exhibiting a well-practiced avoidance response. So the 3 hits of the theory would be being a male, the androgenic hormones, the imposition of very severe stress, and what we do in the laboratory and what you do here in some cases is quite severe. And then thirdly would be the genetic mutations that you're always reading about. But the genetic mutations are a lot harder to use than anybody thought they would be. I can explain that later on. So how do you test this 3-hit theory? I said it's important to have a testable prediction. First, you have to have a mutation, a gene mutation, which is autism related. And what we did was make use of the goodness of Daniel Geschwind, who's a professor at UCLA, and he had a neurally expressed gene whose knockouts, whose mutations are very strongly associated with autism. Another advantage of using this gene is that some papers have reported that the transcripts of this gene are themselves testosterone sensitive. And so that especially excited me because if we have a 3-hit theory where we're looking for interactions, it's not just genetics, it's not just being a male, it's not just early stress, it's the interactions among them. So if here I have a transcript which has already been related to autism, associated with autism in people, in patients, and it's also androgen sensitive, that would be terrific. So that's 1 hit. Then with each of those, we're either imposing a prenatal stress or not, and prenatal stress or not. And the prenatal stress that we chose is an injection of lipopolysaccharide, LPS. So an injection of LPS mimics a bacterial infection. And I chose that because I knew the chemistry of LPS, whereas I didn't know exactly what, what's the other one? The viral infection. Yeah. Poly-IC. I didn't exactly know what that was. And it turns out that it somewhat varies from batch to batch. And so through recognizing my own ignorance, I actually avoided a problem. And so what we're doing is early in pregnancy of a female mouse, we're injecting lipopolysaccharide, LPS, or not. So at this point, we have 8 groups, and a 0-hit group would be a female with no stress and wild type. A 3-hit group would be a male who had been given prenatal stress and who has the genetic knockout. And so what we're doing is we're looking for differences between these groups in their behavior, especially in their social behavior. And I'll tell you one other complexity in just a minute. So here's what we've done. On embryonic day 7, we inject the LPS. Then after birth, we're measuring the first evidence we can get of social communication, which would be ultrasonic vocalizations. The greatest number of ultrasonic vocalizations is on day postnatal 3. And so that's where our data come from. Then we wean the young, and we're getting our tests done as rapidly as possible. First, the open field test. The word anxiety should be in quotes because what it takes to say that an animal's anxious requires a certain amount of faith And I can unbundle that comment later on. But it isn't social anxiety, by the way. It's just non-social anxiety. Then the social approach test, then the social recognition test. You notice that we pack these all together. And then a measure of repetitive behaviors, marble bearing, because lots of autistic kids do a certain amount of flapping and other kinds of repetitive behaviors. But it turns out that the literature on marble bearing is not that great, and our data were even worse. And so I'm not going to present any of those data. And the reason that we were packing these in so rapidly and sacrificing the animals right away is that first, we wanted the molecular biological analyses to be in as close temporal continuity and contiguity as possible. And secondly, because a certain fraction of these animals later on will get epilepsy. And in fact, among autistic patients, I think you probably know that about a third, roughly, of autistic children will get epilepsy. And we didn't want the epilepsy to interfere either with the behavioral test or with the molecular biological test. So here are the data. Here's the ultrasonic vocalizations. You can't quite see that these are way above the human hearing limit, which is what makes them ultrasonic. And the timeline goes like that, and then like that. And we're measuring how many of these are emitted. It turns out that some people think that different kinds of ultrasonic vocalizations, like that one versus that one, mean something different biologically in terms of social communication. But Jill Silverman and I met and talked earlier this afternoon, and she told me that you can play, in some cases, some of these vocalizations backwards, and it doesn't make any difference. And so it's hardly the case that if it's like reading a sentence backwards. Actually, I know someone who can talk sentences backwards, but mainly they don't make any sense. It's like an exotic ability. So for this and for other reasons, like she had animals emitting vocalizations, animals who were deaf, and so they could not have learned social vocalizations, ultrasonic vocalizations, the way you learn a language, because they were totally deaf. So we were glad that we just counted the number of them and we didn't try to make a big deal about which type. These are the last data in this talk that will have the beautiful monotonic decrease in behavior according to 0 hits going to 3 hits. And you can see clearly that, as predicted, the animals with 0 hits had significantly more ultrasonic vocalizations than the animals with 3 hits. And I'll show you all the data here. Always after showing you the data according to the number of hits by my theory, we'll look at all the data. And I think you'll see that the big story is with the extremes. Here's the 0 hits and here's the 3 hits. And in between, I haven't been able to make any sense of it, but the paper's not submitted yet. So if you see some sense in these intermediate data that I don't see, please tell me. So the way this social recognition test operates is that the test animal, here a brown test animal, is sitting there, and you put into that test animal's cage a small plastic cylinder with holes in it, such that the test animal can smell the intruder animal and can hear the intruder animal, but can't touch the intruder animal. And then what you do, the behavior that you're measuring, is the active investigation with sniffing of the intruder animal by the test animal. And what you expect is that you give the test for 5 minutes, then you take the intruder animal out for 15, 5, 15, 5, 15, 5, and you're expecting by this time the novelty of the intruder animal will have gone away. And the inspection by the test animal, because this is not a very large space, the inspection by the test animal will decrease. And then when you put in a new new intruder animal, which here is in pink, that the amount of investigation will go up. And so the data came out just beautifully with respect to our theory, in the sense that with 0 hits, you got the steady decline of social investigation as you go to test 4, and then you put in the new test 5, and all of a sudden it rebounds. Whereas the 3-hit animal had less of an interest to begin with, and absolutely no habituation or dishabituation. The in-between, as you'll see when I show you all the data, once again, the in-between conditions, the 1 hit and the 2 hit, don't lead to any new and startling conclusions. So for example, here you have the extreme of 0 hits. Here you have the extreme of 3 hits with nothing going on. But here, even with 2 hits, which the 2 hits would be the knockout and the prenatal stress, you have pretty good performance in the social recognition test. So the extremes work out, but the intermediate cases do not. Then we had a big defeat. We really predicted that in a standard social preference assay, there would be orderly results with respect to the number of hits. We expect a big social preference, and I'll tell you the assay in a minute, on the part of the 0 hit, very little social preference, and almost social avoidance perhaps, on the part of the 3 hit animals. And what it is a standard three-chamber test in which you put the animal in the start chamber, and the animal has a choice of going either into the chamber where there's another animal versus going into another chamber where there's an inanimate object. There's something there that the animal hasn't seen before, but it's not another mouse. And we didn't get any results at all. And furthermore, when we looked in more detail at all the data, there was no order to the data at all. And so I'm beginning to think that the social preference is very, very sensitive to the way you do the test. And even though we did it in a canonical form, it certainly didn't do anything for our theory. So that's one thing that makes me doubt the theory. Likewise, anxiety. Now, calling an animal anxious requires a real leap of faith, because you're not into the mind of the animal in the way you can be, let's say, in a psychiatrist's office. And most people, if they're going to use the word anxiety-like behavior with a mouse or a rat, they're going to require that you do this open field test where an anxious animal cowers. It doesn't move around very much. When it does move, it's right next to the wall, and it doesn't dare to go out into the lighted part of the open field. Whereas a non-anxious animal moves a lot, it doesn't move just next to the wall, and it does spend a lot of time in the middle. But they would also require, if you really want to use the word anxiety-like behavior, that you do another assay called an elevated plus maze, and still a third assay called a dark light transition. And if all of those assays go the same way, then you might say, yes, this is an anxious animal, and let's use it to develop medicines. But here, we didn't get any significant results, and we were out of time because we wanted to sacrifice the animals for the reason I told you. And so we didn't press it forward to do other kinds of anxiety-based assays. So the purpose of this work was to start with the behavior and test the 3-hit theory, but then to go into the brain and look at gene expression in the brain, and then as part of Sarah Schaafsma's postdoctoral training, to do so-called epigenetic testing, which I'll explain in just a minute. And what we did was, since we're dealing with stress, was we examined quantitatively lots of transcripts related to stress systems in lots of limbic structures. And Sarah insisted, because she'd done previous work in the Netherlands on left brain versus right brain, on dividing up the left hemisphere in the limbic system from the right hemisphere. And I didn't see why we were doing that. I thought it was a waste of time, but it turned out I was wrong. Because of all of the genes we tested, the only one where we got significant results was corticotropin-releasing hormone receptor type 1, messenger RNA. And relative to the 0-hit condition, in the left hemisphere but not the right hemisphere, the left hippocampus but not the right hippocampus, we got significantly decreased CRH1 messenger RNA in the 3-hit group compared to the 0-hit group. And that did not obtain over here. And if I show you all the data, once again, here, the significant results are over here, not on the right side. The 3-hit, smaller, less messenger RNA than the 0-hit. And there is a tendency here, I think that's in the females, like the wild-type female with stress and the knockout no-stress female. There's a tendency here for monotonic decline, but the only thing that was strongly significant in our view was the 0-hit versus the 3-hit. Now at that point, we wanted to say, we do have the basis for doing epigenetic studies. If we didn't have any messenger RNA results at all, we wouldn't be able to do epigenetics. But what we wanted to do now to test in the left hippocampus was to do studies to try to figure out the reasons that the 3-hit animals have less of this messenger RNA than the 0-hit. And at that point, we had what at lunch with the postdocs and the students I called the Goldilocks choice. Remember Goldilocks and the bear, where some porridge is too hot, some porridge is too cold, and some porridge is just right. And we had 3 choices of doing epigenetic work. One is DNA methylation, which is a way that the environment can influence gene expression through the life of an individual without changing the DNA itself. A second is modification of proteins that coat the DNA. There's no such thing as naked DNA. And the third is microarrays, non-translating RNAs. And the Goldilocks choice that I made was that for my purpose as a neuroscientist, I thought the DNA methylation is too simple to be the basis of all of these complicated social behaviors. And microRNAs was too complicated because there's over 5,000 of them. So I thought if I got one choice which is too simple and one choice which is too complex, I'll choose the choice in the middle, the porridge that's just right. And so in that sense, we have a limited number of histone proteins and a limited number of epigenetic modifications which we could study. And another reason for doing the histone work, histone protein work, is that one of the world's experts is about 30 meters from my lab. So I could go and ask him advice, I could get reagents from him, he could tell me where to buy stuff, and that was very good. So what we did, the idea of this is that upstream of the CRHR1 gene is the so-called promoter area where proteins will bind that will either wrap up the DNA and keep it from being expressed, messenger RNA from being expressed, or it will open up the DNA such that transcription factors can go in and turn on that gene. And the promoter area that we chose, 5' to CRHR1 transcription area, has a binding site for SRY and a binding site for NF-kappa B. So remember back to one of the first slides, the SRY is the protein that's centrally involved in sexual differentiation, and NF-kappa B is a stress-related signaling system. So we thought if our 3 hits theory includes sex and stress, choosing a promoter area which should respond to both of them, which reasonably could be expected to respond to both of them, is a good thing to do. And we got some startlingly positive results, but then we also ran into a reversal. Take a look at histone 3 acetylation. Clearly, you've got more acetylation, which is a pro-transcriptional mark, more acetylation in the 0-hit group than in the 3-hit group. And since we're trying to explain more messenger RNA in the 0-hit group of animals, the fact that this green bar is bigger than this red bar is good for business. Likewise, histone 3, lysine 4 trimethylation, the green bar is significantly bigger than the red bar. And so those could be taken to lead to a line of causal evidence which would say that because of histone acetylation in the promoter area of the CRHR1 gene, there's more CRHR1 expressed, and that has behavioral effects with respect to response to stress, just like the doctor ordered. But then we did the mistake of doing too much work. And this is histone 3, lysine 27 trimethylation, and it's just the other way around. This is a repressive mark. These marks, these transformations of histone proteins, are uniformly known to increase transcription. This is uniformly known to repress transcription, and yet we have the same pattern of data. You'd almost think as though we mixed up the samples. And so the answer is that these are very much in favor of the theory that I've been talking about with response to sex and response to stress, and this goes absolutely against it. And I don't know how to explain that. I think I've got a data slide with all the data, and once again you'll see that the big story in each of the histone modifications is the extremes, but in between there's nothing like a monotonic decrease as you go from the green bar over to the red bar. And why there's not a monotonic increase, I don't know. When I talk about statistical interactions, the statistical test of interactions among 3 factors, I know what I mean. But what does that mean in terms of biologic reality? And in what do those biologic realities mean in terms of these intermediate groups, like right in there, is something we just don't know. So at that point, we had a thin line of causal evidence going from epigenetics to gene expression to social behavior, and at no step in that did all the data pull in the same direction. Always we had some kind of contradictory data which I can't explain. So I began widening the net, and I thought, what are the molecular endocrine sources for social deficits due to androgen action? Because that's what we're talking about. The most seemingly likely source would be the androgen receptor, which is on the X chromosome, and since the male only has one X chromosome, a mutation there would lead to some kind of defect. but there's no evidence regarding that so far. Related possibilities in the androgen system would be in the androgen receptor promoter, androgen receptor enhancers, or the chromatin overlying androgen receptor gene. And since testosterone is aromatized to estrogens, you have the corresponding set of potential explanations in the estrogen system as well. And as far as I know, those have not been tested. So what we did instead was to look for the largest number of possibilities, to increase our catchment, so to speak, by doing a microarray. Many of you know that in order to demasculinize the brain of a female laboratory rat or laboratory mouse, the critical period for that defeminization or masculinization action is right around the time of birth. And so here's a rough sketch of the sensitivity to testosterone as far as sexual differentiation is concerned in the laboratory rat or the laboratory mouse. And what you may not know is that if you castrate an animal, a neonatal rat or mouse right in here, that animal will behave like a female for the rest of its life. And if instead you give an injection of testosterone to a female laboratory mouse or rat, that female will behave like a male the rest of its life. It won't do the female sex behavior. Instead, it will run around trying to mount other females. So what we wanted for our microarray study was to have it late enough on the day of birth that it would be after the early genomics of testosterone or aromatization of testosterone to estrogen. And the way we calculated it, we were about 2/3 or 3/4 of the way through that period as far as area under the curve is concerned. But also, we wanted to have this period where we were sacrificing the animal for the microarray and harvesting messenger RNAs to be causal to sexual differentiated behavior, so not the result of it. So we couldn't wait too long, because then the animal might begin to show sexually differentiated behavior, and our RNA results would be the result of behavior, sex differences in behavior, rather than the causation. So that was our methodology and our reasoning, and here are the results. So these are log scales, log scale, log scale, log scale, log scale. The male's always on the y-axis, the female's on the x-axis. Here's the preoptic area, which is a cell group which is essential for male sexual behavior. Here's the medial basal hypothalamus, a cell group which is essential for female sex behavior. And the red dots, out of 11,000 genes, more than 11,000 genes, the red, or they look purple here, purple dots are where the male transcripts are present in greater numbers than the females for any given gene. Each dot is a gene. And the yellow dots are where the female transcripts are present in greater concentration than the male. And in the preoptic area, an amazing 8% of the transcripts had some kind of significant sexual difference. I didn't expect it to be that large. In the medial basal hypothalamus, it's only 0.9%, but when you're dealing with over 11,000 genes, 0.9% is still more than 100 genes, so it gives you plenty to think about. And I'm going to talk about the amygdala in a couple of minutes. Now, these transcripts, which are sexually differentiated in our experiment, had virtually every kind of biochemical category. There were transcription factors, there were elongation factors, there were translation factors, kinases, phosphatases, and so a couple of other genes that I'll talk about. And so we had to pick one. I couldn't study all of them. And here's the one we picked. Or I should say, here's the two that we picked. Connexin 36. Now this gene yields messenger RNAs, which will then yield connexin 36 proteins. And these proteins form themselves into little barrels, little six-sided barrels that are in the nerve cell membrane. And they form a hemijunction in a neuronal membrane, which, if paired to another neuron, which also has the hemijunction, has a little tunnel, sort of like the tunnels that go from Egypt into the Gaza Strip. And through those tunnels can go charged molecules, which essentially make electric synapses, which can be very powerful. They're certainly very fast. And if you're looking for ways to spread electrical excitability fast, then having a lot of connexin 36 in your cell group is the way to do it. We used as a control connexin 43 in astrocytes, coupling to developing neurons into other astrocytes. And we didn't think that would have the same biology. So it's easy to get significant results with our experimental design. This is the work of Lars Westberg, who's now back in Sweden. And so if you see here, in the medial basal hypothalamus with respect to connexin 36, You have a significant sex difference, neonatal males compared to neonatal females, and you also have a significant size difference. The sex difference does not obtain for our control, connexin 43, and the size difference, the age difference is exactly the opposite. Here the adult males were less, and with connexin 43 it's a lot more. And we felt that we had to do a lot of cell groups because we didn't know where the results were coming from, but our main focus had to be on the amygdala. And we're looking for data that would make the amygdala, quote, supersensitive. And now the connexin 36 in the neonatal male is significantly greater than in the neonatal female, and that difference does not hold up for connexin 43. The neonatal female and the neonatal male have about the same amount of messenger RNA for connexin 36. So we think, and maybe wishful thinking, that expression of connexin 36 early in the life of a male rat is likely to lead to more of a supersensitive amygdala than the female, and that might be part of what I was talking about at the beginning of the talk. So wrapping it up, this is wrapping up the real talk, and then good. We have time for the epilogue, where I talk about things that we hope to do just now, because the best way to open them to criticism is to talk about them. So wrapping up the real talk, I claim that neuroendocrine and molecular endocrine literature, which a lot of neurologists don't know that much about, and techniques offer new approaches to a major feature of autism spectrum disorders, the sex difference. A difference that I thought was going to be really easy to wrap up, but I'm 7 years into the mission now. We have evidence that increasing the number of hits increases the behavioral consequences in mice. Some of the evidence, both behavioral and molecular, is very strong, but in each case we had some counterindication that we haven't explained. Based on our microarray study, the numbers of genes transcripts involved in behavioral sex differences could be very large. And I can relate that to the literature in general. 10 years ago, people funded, for example, by the Simons Foundation, were looking for the autism gene. And so we had neuroligands, we had SHANK3, we had catnap2, the one that I used. And then there got to be more, and then there got to be more. And if you look at the literature in the last 2 or 3 years, it's evident that there are hundreds. Furthermore, from the work of Christopher Walsh, who's a professor at Harvard Medical School, he says that some of these de novo mutations actually occur in some neurons in the cortex, but not in others. So that you get this salt and pepper expression of gene expression in the cortex, which makes me think that the purely genetic approach is going to be very difficult to turn into something which is really actionable from the point of view of real doctors working with real patients. Questions from our own work. Our new social approach data were the opposite of what we expected. That's not the social discrimination, it's the social approach. And furthermore, the molecular phenomena in the left hippocampus and not the right have very few precedents. I mean, Sarah Schaafsma, my postdoc, was right, but why she was right is something we still don't know. So at this point, I felt some dissatisfaction because it had taken us 3.5 years of hard work to do the catnap2 study, the Sarah Schaafsma study. And then when I do the microarray, I've got like hundreds of genes that I could look at, more than 100 in both the hypothalamus, and in the amygdala and in the preoptic area. And I don't have time to deal with hundreds of genes. So I began to think strategically, the way I have all of my career, which is to try to look for a clean experimental situation in which either the behavior and the likely neural mechanisms are very simple, and I can figure them out within one lifetime, or where I'm beginning at the beginning and I'm dealing with a tabula rasa situation. And so I started thinking about the fact that prenatal children, children born early, have an increased risk for neurodevelopmental disorders. And I thought, well, there are neonatal intensive care units, and we have lots of those in New York City, so I'm going to go visit my friends and watch babies. So I signed all the appropriate forms and would wear the appropriate clothing, and I would go and watch these babies the way an ethologist looks at animal behavior. An ethologist is a person who wanders out into the woods and looks at natural behavior of non-experimental animals in their natural environment. And the natural environment of a prematurely born infant is the neonatal intensive care unit. So at Mount Sinai Hospital and at Roosevelt Hospital in New York City, I'd watch these babies for hours. And I thought, wow, if these babies are at increased risk for neurodevelopmental disorders, we also have them here in a perfectly controlled condition. As soon as those babies get to see their mothers and growing up in the real world, all the bets are off. But these babies have to be rigidly controlled in order to keep them alive. Some of them are born as early as 30 weeks of age. And so, as I was standing there watching the autonomic measures of these babies, the heart rate, the breathing, the degree of oxygenation, I thought, what's the earliest possible measure of a social response? And things are going to get a bit crazy here for a minute, but see if you agree with this. The earliest possible measure of a social response that I've been able to think of is that when the baby is in close contact with the mother, if the mother's autonomic nervous system changes, the baby's autonomic nervous system will change. In other words, it's going to be brain-to-brain communication, but not cortex to cortex, but autonomic nervous system, really primitive, to autonomic nervous system. So we have two kinds of projects going on now. One is in the neonatal intensive care unit, where we have 20 dyads who are signed up for kangaroo care. And kangaroo care, which is good for the babies anyway, is where the mother and the baby are opposed to each other, chest to chest. And what we have is we're measuring EKG from the baby and EKG from the mother with very high temporal resolution. And we're making the prediction, the simplest prediction would be, as I just said, and we have the data, that if mom's heart rate goes up, baby's heart rate will go up. Or let's keep our mind open. Maybe this communication goes the other way. If baby's heart rate goes up, then mother's heart rate go up. But if we were to limit our entire project to that simple arithmetic prediction, we'd probably be begging for failure. So I have a friend who's a professor of mathematics at the University of Virginia. He's been a visiting professor in my lab. And we're sending all the data to him. And we're not just looking at simple rates. We're also looking at heart rate variability, shared mutual information, shared entropy. He's got a wonderful statistical imagination. And so we'll keep going, knowing that some dyads may be great communicators and other dyads may be very poor communicators, hypothesizing that one early test of social capacity of the baby may be to communicate heart to heart, literally heart to heart, with the mother. And if we could achieve that, the reward would be very large, because we could do it then mathematically. We could establish standard curves the way you do in enzymology, and then we would know that a baby who's off the standard curve, especially a boy baby, in the wrong direction, that is too little response to mom, may be a baby where palliative measures should be taken right away. That's one of the two. And I'm not a clinician, so I just have to be catalytic. I have to come up with ideas and criticisms and encouragement because the clinicians lead a hard life. The other project that we have going is actually in utero, where Catherine Monk, who's a professor at Columbia, has recordings with the sonogram from the baby and EKG from the mother. And we're doing exactly the same thing as I just told you. All of the same analyses, and we have the data, and Dan is working on them, but Dan leads a complicated life too, so I can't show you the results yet. So as I said, I'm not a clinician. But again, I want to focus on the earliest events, and this neonatal intensive care business I just told you about would qualify. We've got the baby from the get-go. The other way is to get some of the critical cells from the get-go. And you may remember, I hope I remembered to say, that at the beginning, when I talked about those nucleus gigantocellularis neurons in the lower part of the brainstem, I wanted you especially to memorize those because they have androgen receptors and because they're critical for arousal of all sorts. And so that was step one of the three-hit theory, the NGC cells of the lower brainstem. And so we've got those cells now. And what we do, I want to study effects of compounds and treatments on those cells that are thought to stand in for the kind of treatments that could predispose to autism. So those cells are born, here's an embryonic mouse, those cells are born on day embryonic 10. And we sacrifice them on day embryonic 12 because they're well established in their final resting place at that point, but they haven't sent out neurites yet. If we waited until they sent out neurites, then we'd be cutting the neurites and the cells would probably die. And so we dissect out the medial hindbrain to get those NGC neurons. We dissociate the cells enzymatically. We spin them down. And then we do a trick, which I learned when I gave a talk at the University of Illinois. There's a brilliant biomaterials guy there by the name of Brendan Harley. And when I saw in his research description, he wasn't just doing cultures of cells, bone cells, tendon cells, the kinds of things that a biomaterials scientist wants to grow. Bone cells, even glial cells, but he'd never done neurons. And he's constructing these hydrogels, and what you do is you take gelatin molecules, or what Brendan does, is take gelatin molecules, and he does what he calls functionalizing them with methacrylate moieties. And then he takes hyaluronic acid, that should be photoinitiator, and certain survival factors. And what we do is to follow his we do our dissection, and then we follow his procedures in collaboration with him. And then I expose them to ultraviolet light, and the ultraviolet light provides the energy for these reactive groups, the methacrylate groups, to cross-bind to each other so you have a three-dimensional gel. And the idea is that we have our neurons growing in little tunnels, not just out in the air, but growing in little tunnels, hopefully to be a little bit more like the brain. Now, what we haven't done yet is to put in glial cells. I wanted the NGC cells to be all by themselves, but probably it's not as smart as putting in glial cells, because that's what nerve cells expect to have. So I give the ultraviolet light, then we incubate in neurobasal media, and we ask, can we get these cells to survive? Can we get them to grow? And would they be a quantitative measure in confocal microscopy of the harm to the growth process that might eventuate if we give them certain treatments? And so here's what we've done so far. This is the work of Ana Maria Marinos in the laboratory, who's a biochemical genius. And here's an example of an NGC cell, or shall I say an NGC-like cell in the 3D matrigel. And this has been 11 days in vitro, and you can see that it's sent out processes very nicely. We hope that we're building little circuits in there. And the purpose of that is we can do all of this quantitatively. We can then look quantitatively at the effects of hormone administration on these cells, environmental endocrine disruptors, anoxia and other disturbances related to cell metabolism, effects of environmental poisons and their remediation. Lead would be an example, which is in the news right now, but unfortunately, there's an infinite supply of these things that we can experiment with now that we have the system set up. So I gave about 80% of the talk or 90% of the talk based on two long experiments, the CatNap2 and the microarray experiment. But actually, I'm sort of dissatisfied with them, and I thought it was time this is the first time I've talked, I think, in public about these new things that we're trying to do, and which could be criticized, which no doubt you will do right now. Thank you very much.
This is a question about your epilogue and the autonomic coupling. And as you know, in DSM-5, sensory processing is part of the criteria that's been added to identify a diagnosis, either hyper or hyposensitivity. So when you're conceptualizing sort of heart-to-heart communication, that's still mediated by somatosensory systems in the brain. And so in terms of sensory sensitivity, are you conceptualizing a kind of over-aroused system or an under-aroused perceptual system of the mother's body by the baby?
Yeah. I think I'd be disposed to predict that that's over-aroused, hyper-aroused. So I think you started out talking about DSM-5, right?
Yeah.
Yeah. Okay, so first I'll answer a question about that, and then we'll get to your direct question, because I've distracted myself by answering part of your preamble first. Is that I consider these borderlines in diagnoses to be artifacts, that they're necessary for legal and social procedures, but basically what they're doing is they're imposing straight lines on continua. And so a person whose symptoms are just above the line is essentially like the person whose symptoms are just below the line. But for purposes of reimbursement and so forth, you have to have a name to apply. But from the point of view of creative research, I don't pay any attention to them at all. Now, in terms of the prediction that we're going to make about baby and mother, I would guess that in an autistic baby, a baby that is destined to have a life with symptoms of autism, that there would be a hypersensitivity. But does that necessarily mean that there will be an autonomic response? I don't really know. because you focused in your question on the sensory side. But to get the data, we've got to get back out through the autonomic nervous system, and that's a more complicated calculation than you would think.
Right. Ami Klin is doing third trimester 4D ultrasound of responses to babies before they're born to the mother's voice to see whether in siblings of kids with autism they can predict a phenotype prenatally.
I hope they can. So he's a genius, and I think that's a good idea to do. If you want further data along the lines of evoked potentials, look at the work of Ira Cohen, who's at the Staten Island unit of the State University of New York, who has published work in that area. Yeah.
Thank you for your talk.
Yeah.
Can you speak to the sex chromosome disorders and how that would fit into your model, three-hit model, in terms of Klinefelter syndrome, Turner syndrome, all, the disorders that have extra X or Y chromosomes?
Honestly, I don't know.
Okay.
In the first place, I can never remember which is which. I haven't dealt with. I cross-registered into medical school when I was at MIT, but the last time I've encountered those things was back in medical school. And but what I can tell you is that things don't always come out the way you would think. For instance, the XYY, you would think would be hyper-aggressive men. And at first, people did think that, like Siamese fighting fish, XYY individuals would be more aggressive, but then when they did the study properly with another control group, it happened to be a prison population, the data washed out. I'm acquainted enough with the complexities of chromosomal localization and chromosomal crosstalk in the cell nucleus that I can clearly see room for trouble when there's an extra chromosome, but I can't line up, as your question intended, I can't line up the nature of that chromosomal disorder to the nature of the behavioral phenotype.
Yeah, have you thought about using some of the rodent models where you can separate out the sex differences, the genetic basis of sex differences from the hormonal? I think they do gonadectomies, and I know I've seen these models before where you can separate out, because there's the X chromosome effect,
Yeah.
which is purely a chromosomal genetic, probably leading to epigenetic effects versus the hormonal effects. Have you thought about trying to separate out those two parts of the question?
One of the reasons I haven't tried to separate it is, in the first place, I don't have money for that size of mouse colony. And secondly, Art Arnold, who's a professor at UCLA, is so much better at it
That's where I think
that I'd probably end up calling up Art and asking him what to do. Honestly, despite the fact that I admire his brilliance and the guts that he took to go against the prevailing theory is partly my fault, about sex differences in behavior, is that for all of that work that he put in and his consortium put in over a period of 7 or 8 years, when you look at the percentage of causation of behavioral sex differences, it's still far on the side of the hormones. And that's why I didn't blanch at all when at the beginning of the talk, and I gave the first explanation, namely the testosterone, I said I'm going to talk mainly about this, because doing it that way, I can do easy work and explain 80 to 90% of the data, whereas with Art's approach, I would do a lot of work and only explain 10 or 20%. And so I'd rather do the former. I think Art's right. Every cell has a sex. But how much does it bear on the things we care about as experimentalists and clinicians? I don't know. I'm not so sure about that. And I think when you look at Chris Walsh's paper, you'll see that there's going to be plenty of room for complexities we didn't need.
Can I ask you a question? So I've always been intrigued by this issue of sex differences in autism as well. And so up until recently, there hasn't been a good explanation. You've offered some. The other explanation that seems to be coming out is that females do really have a protective effect, in the sense that if you look at the copy number variation data and you look across a large population, you need to have more copy number variance in females diagnosed with autism than males. If it takes 5 for a male, you need to have 10 for a female, just to throw numbers out. And so for whatever reason, females are able to absorb more of a genetic hit than males. I mean, at least that's the notion.
Yes.
And I just wonder what you think about that.
I think that's true. And if I were to reword exactly what you said, it would be that the females have to have a larger genetic hit in order to have a certain pathologic status of their behavior. And so first, I would say that where those hits are is still completely up in the air. There's nothing specific about it. And secondly, I think that there probably is the chance of a diagnosis bias. That is, if you have a certain pattern of less than average social behavior, if it's a boy, you say, oh, this kid has autism, and especially if you're a teacher who wants to get you out of her class or his class, that diagnosing with autism and getting them in New York, we call it District 75 for children with neurodevelopmental disorders. You want to get the kid over to District 75, where if it's a female and she's slightly aloof, you would say, oh, how graceful. It's part of the natural individual variation. So by the way, I think that's a good way to do research, is what Eric London, who's an expert in autism and a clinician, is doing in New York, is that we have one school where all of the, in the South Bronx, where all of the District 75 classrooms are children with autism. And just now we're getting money in order to study those children systematically during the school day.
I noticed on your method slide for the rodent model that you had put a little star next to the prenatal stress, citing Tracy Bale's work.
Yes.
And I was wondering if you had ever thought about, instead of doing the maternal immune activation as your prenatal stressor, using her chronic variable stress paradigm or some other stress paradigm.
You think her what?
Her chronic variable stress paradigm?
I'm afraid of that. In other words, my friend Bruce McEwen, who's a stress expert, did for a long time a stress that we call shake, rattle, and roll. And what that meant was take the animal out of its cage, and it's swinging around in a net, shaking, and there's noise, really vicious New York radio stations playing at it, and it's being tossed around. And the sensory pathways that register shake, rattle, and roll are so complicated that by definition, as a person who has a certain amount of neuroanatomical expertise in my background, I don't know how the hell it's working. And so when I looked at Tracy's, I thought it was very clever, and she does wonderful work, because the animal cannot predict from day to day exactly what's going to happen. But I have organic chemistry in my background, and so when I had a simple chemical like glycosalicylsaccharide and maternal infection would predispose to autism, I had to go for the simple defined chemical route that we give just once, rather than the more complicated Tracy Bale route.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.